Hello everybody and welcome to our video today where we are going over biology, chapter 1, the cell. So let's get started. I know I don't have my glasses today and I can't find them and I wanted to search for them, but you know what? MCAT doesn't wait for anybody, so we just gonna have to power through today without the glasses. So first we have, like we said, bio chapter 1, the cell. So let me just move this over here so you can see a little bit better. All right, so first we have the parts of the cell. So, let me just maybe stand on this side. There we go. Okay, first we have the parts of the cell. So we have, in prokaryotic cells, we have plasmids, which are the part that carries the DNA in prokaryotic cells. In prokaryotic cells, we also have the nucleotide region, which is the DNA region in prokaryotic cells. So we have the nucleolus, which makes ribosomes in the nucleus with no membrane. Then we have peroxisomes, very important, and they collect and break down material. Then we have the rough, these are again very general, just brief things you should know about these parts of the cell. You know, they do a lot more than just, you know, this. So I would say if you aren't familiar with these parts of the cell, definitely look at another video for that. But just in general, just a brief overview. Yeah, so now we have the rough endoplasmic reticulum, which accepts the mRNA to make proteins. We'll go over that in more detail in another chapter. We have the smooth endoplasmic reticulum, which is kind of a detox center, and it makes lipids. That's important. We have the Golgi apparatus, which modifies and distributes proteins in eukaryotic cells. Then we have centrioles, which are a group of microtubules that pull apart the chromosomes. We're going to get into that in the next chapter as well. We have lysosomes, which is kind of a demolition and recycling center, and they're made by the Golgi, sorry. Then we have the mitochondria, and mitochondria are important because they have their own genes and they replicate independently by binary fission. There's a couple other details to mitochondria, and we definitely will look at this in later chapters as well, but just, you know, be able to remember this part about mitochondria for now. And then we have the cytoskeleton, and this part can be a little more confusing, so just make sure you know these differences. So we have, in the cytoskeleton, right, we have microfilaments, for example, actin. And that actin specifically interacts with myosin for muscle contraction. So that's an example of a microfilament. Then we have microtubules. And now the microtubules, for example, tubulin, is a pathway for motor proteins. Then we have intermediate filaments, which kind of anchor things to the organelles. Anchor the organelles to the cell. And for example, that's keratin and desmin. So these three differentiations of the parts of, or the different types of things you can find in the cytoskeleton is important. I have seen, you know, questions on that specifically. So let's look at this next. Basic differences. I'll just move this down a little bit. Between, oops, sorry about that. The differences between, okay, have to be like that for now. So the differences between eukaryotic cells and prokaryotic cells. So eukaryotic cells have membrane-bound organelles, while prokaryotes do not have membrane-bound organelles. Eukaryotes have a nucleus. They have their electron transport chain, which we will go into another chapter. They have that in their mitochondria. Eukaryotes also have large ribosomes, and eukaryotes reproduce via mitosis. We go over the types of reproduction in the next chapter, but just note that difference. Now you have prokaryotes, which, as I said, have no membrane-bound organelles. Their electron transport chain is in the cell membrane instead of the mitochondria membrane. Prokaryotes have small ribosomes. They have no nucleus, and they reproduce via binary fission. Okay, and they also can have genetic recombination, which we will go over here. So now, let me step over here. So prokaryotes can have genetic recombination through transformation, conjugation, transduction, and transposons. So we have those things written here. So via transformation means that the genetic information from the surroundings are taken up by the cell. Then we have conjugation, which is a transfer between two bacteria using a bridge plasmid. Okay, so the F plus and F minus, you'll see that frequently used here. That's, and that is transferred from one bacteria to another. Let me just make sure we got all those parts here. Yes, okay, perfect. Then we have transduction, which is the transfer of genetic information using a bacteriophage vector. And then we have transposomes, which are genetic elements that can insert and remove themselves from the genome. So these four ways is how prokaryotes get genetic recombination. Recombination. But then when they reproduce, that is by binary fission, okay? All right, so our next thing that we're gonna look at is, let's take a look here. Next, we have bacteria. So bacteria can be obligate aerobes, obligate anaerobes, facultative anaerobes, and aerotolerant anaerobes. And so each of these things basically describe what the bacteria needs in order to survive. So there's a couple of different environments where bacteria will succeed or thrive, I should say. So we have obligate aerobes, which means this bacteria would require obligate aerobe oxygen. So it would need oxygen. Obligate anaerobe means that it has to have no oxygen. So that means it would die in the presence of oxygen. Then we have facultative anaerobe, which toggle between aerobic and anaerobic. Then we have aerotolerant anaerobe, which means that this bacteria would not use oxygen, but it can tolerate. So it won't die in the presence of it, but it also doesn't need it. Following this, we have gram-positive or gram-negative bacteria. And so what this means, gram-positive bacteria would have a thick wall composed of peptide glycan. And when you are doing a gram stain on this, it would come out purple because of the thick wall. So this would be gram-positive bacteria. Then we have gram-negative bacteria, which would have a thin wall with an inner and outer layer of lipids, and it would be pink. Okay, so purple would be gram-positive, pink would be negative. Purple is the way I think of it is purple is a darker color and it's gram-positive, purple positive, and it has a thicker wall. Whereas gram-negative, it wouldn't have as much of that stain in it. It's a thin wall, which means it would be pink. Okay. So the final part of this chapter is viruses. Now this is a hefty part, so make sure we are understanding this. I know it took me a while to understand some of these concepts. So viruses have to contain a capsid and genetic material, and they also require a host cell. So bacteriophages are viruses that target bacteria specifically. Then we have viruses can be composed of RNA or DNA, single-stranded or double-stranded DNA, okay? So for RNA single-stranded, if you have a single-stranded RNA virus, you can have it be positive or negative. So if it's a positive RNA single-strand virus, it can be translated by the host. So positive just easily is translated the virus, okay? Now it's translated by the host. But if you have a negative RNA single strand, that means that this virus requires a template strand to be synthesized using RNA replicase before it can be translated by the host. Then we have retroviruses, which are single-stranded RNA viruses from which a complementary DNA strand is made with reverse transcriptase and then is integrated into the genome. So this is an important type of virus, and it's a little bit different. So you have a single strand of RNA, and from that RNA, the host cell actually creates a complementary DNA strand, which is reverse transcriptase. Because when you're transcribing, right, you take the DNA and you're creating RNA. But this type of virus actually, forces the host cell to use reverse transcriptase and create a DNA strand for the RNA virus, and then it is integrated into the genome, the host cell genome. So now we have, what else do we have here? We have bacteriophage life cycle. So we have lysogenic and lytic cycle. So the lysogenic bacteriophage part of the life cycle, as the virus integrates into the host genome and replicates. So lysogenic, you just are integrating into genome, replicating, and you know, you're active. But then you have lytic, which is when it produces virions, which are virus particles that lyse everything that is considered very, I can't say that word, virulent, sorry. So that is the lytic life cycle. I'm just trying to see here if we used any other words that, okay, so we have prions also, which are infectious proteins that trigger misfolding. And a lot of the time they convert alpha helixes to beta sheets. That is prions, infectious proteins that trigger misfolding. And then we have virioids, which are plant pathogens, and they're small circular RNA that can turn genes off. Whew, sorry about that. Okay, let me just make sure here that we covered everything. I believe that we have. Let me see here. Yes. And like I said, this is the basics of a lot of the biology that we're going to be using, and, you know, looking at different parts of the cell and things like that. So these are just very, you know, hopefully a summary of things that you've looked at before, and just a quick refresher before we get into, you know, more complex topics in these chapters. One thing I did forget to mention was for viruses, the 2 steps that they basically go through to, or maybe you couldn't see that, maybe it's because it moved. So for viruses, the 2 steps that the viruses go through to infect are actually, the first step is attaching, the virus is attaching to the host cell. And then the second step is when it infects the host cell by injection. I just feel like there's one part that I forgot to mention. Just give me one moment here. Yes, okay, so for the lysogenic cycle, it integrates the virus, yikes. The virus integrates, for the lysogenic cycle, the virus integrates into the viral genome of the host cell. Sorry, integrates the viral genome into the host cell's genome. But then it will just go through normal cell activity. And then at some point it will become active and begin to lyse the host cell and release the viral particles. But you could, for a period of time there, which is unknown, the period of time, that it will just have normal cell activity for the lysogenic. And finally, it will, as I said, remove the repressors and then start the lytic cycle. So the lytic cycle is, you know, the act of lysing the host cell, whereas the lysogenic cycle is integrating it at first, integrating the viral genome at first, and continuing with normal host cell activities, and then at one point in time, removing the repressors and starting the lytic cycle. And then from the lytic cycle is when you're taking you're transcribing the viral genome, you're replicating it, and then you are lysing the cells. So that's just an important clarification I wanted to make. So yes, thank you for coming to this video today, clicking on it. And we'll go into the next chapter shortly. And I hope you all have a wonderful day, and put any questions, comments, or concerns below.